So first of all, the really important thing to realize about non-membrane bound organelles is that because they're non-membrane bound, that means that they are allowed to be in both prokaryotes and eukaryotes, because the definition for a prokaryote is that it can't have any membrane bound organelles, but these are non-membrane bound, so they can be in both prokaryotes and eukaryotes. They are found in both prokaryotes and eukaryotes. So it is not incorrect to use the term prokaryotic organelle, even though most people might think that it is wrong. Most people say, oh, prokaryotes don't have organelles. That's not true. Prokaryotes do not have membrane-bound organelles. Okay? So ribosomes are the first ones. I know you guys read about ribosomes a few days ago, but I want to talk about them. Ribosome's job is to create a polypeptide using a template from a mRNA molecule. So it creates polypeptides using a template from mRNA. Ribosomes are made out of rRNA and proteins. Does anybody remember where the rRNA and protein are produced that make up ribosomes? Part of the nucleus? The nucleolus, yes, that's where those things are produced that make the ribosomes, and then they travel out through the nuclear pores and then float around in the cytoplasm. There are two parts to every ribosome. There's what's called a large ribosomal subunit and a small ribosomal subunit. And an interesting thing about this is that they don't actually join together until there's mRNA around. Okay, so in the cytoplasm of every cell, there's a whole bunch of the large and small subunits floating around, and there are no complete ribosomes. Right? The only time you have a complete ribosome is when it's attached to mRNA and it's currently making a protein. After it is finished making the protein, it detaches and turns back into its small and large subunits. Okay, so only attached together when joined to mRNA. There are two types of ribosomes. Okay, eukaryotes have a type of ribosome called the bound ribosome, but both prokaryotes and eukaryotes have what are called free ribosomes. We say free ribosomes are free floating in the cytoplasm. We talked about this word yet, cytoplasm? Have you guys read about cytoplasm? You're going to see two things. In fact, this is a very commonly sort of misused term. The term cytoplasm means the space in between the outside of the nucleus and the cellular membrane, right? So anything that exists in that space that's between the nucleus and the cellular membrane is called the cytoplasm. Okay? A lot of times people think that the cytoplasm is the name of the fluid-like substance that everything floats around in. That's actually got another name. It's called the cytosol. But a lot of times people use those two terms interchangeably, even though it's not technically correct, because there's cytosol inside of the nucleus as well, and that area is not called the cytoplasm. If it's inside the nucleus, it's in the nucleus, right? Whereas the cytosol is found inside the nucleus, it's found in the lumen of the ER, all those places. Yeah?
So on the test, would we have to be really specific?
I mean, no, just because, people make that mistake all the time. It's like one of those things that people have made the mistake so many times that it kind of becomes accepted.
Like effect and defect?
I'm really trying to get good about affect and effect, so like I'm still gonna try to get those right. I'm talking about like regardless and irregardless. Like irregardless is not a word, but people say irregardless of it not being a word. Right? Free ribosomes are free floating in the cytoplasm. And because they're free floating in the cytoplasm, we'd say that they're found in both prokaryotes and eukaryotes. Both prokaryotes and eukaryotes have them. What does a free ribosome make? It makes a protein, right? But these are proteins specifically that will be used within the cell. Okay, and so we say these are proteins for intracellular use. They produce proteins for intracellular use. Okay, this is a good time to start getting familiar with this prefix intra, right? The prefix intra means within, right? The prefix inter means between. Okay, so they're very similar prefixes, but they mean two totally opposite things. Intra means within the cell, inter means between cells. So intercellular means between two cells, intracellular means within the cell. Okay, this is very similar to the way that like intramolecular forces versus intermolecular forces, if you've been in chemistry or if you've just been alive recently, that's the same as like the internet and the intranet, right? So people talk about intranets like within a building. So if I wanted to like go over to that computer and print to like one of the nice printers in the business department, I could do that using our school's intranet, right? But if I wanted to like send an email to my mom in Ohio, I would use the internet, which connects between different areas, right? So within, intranet, internet, between areas, right? Well, perfect. Now you know. Knowing is half the battle. Intranet is within a building. So like your home network is an intranet. All right. So what else was I talking about? Okay, so the other ones, bound ribosomes. Bound ribosomes attach to pores on the rough ER. And therefore, they're only found in eukaryotes because prokaryotes don't have rough ERs, right? And these are going to produce proteins for export. They'll also produce proteins to be used like within peroxisomes and lysosomes and stuff, but for the most part, you can be sure that they're going to be used for export the vast majority of the time. Okay? So this is all ribosomes. Interestingly enough, there's actually no difference between free ribosomes and bound ribosomes. They're the same small and large subunit. The difference comes in the mRNA. So the mRNA will attach to the ribosome and say like, hey, this thing needs to be exported from the cell. And so if it's got that little snippet of information on the front of the mRNA, when the ribosome's attached, they'll go over and migrate towards the ER, and they'll attach to one of the pores so that the protein gets produced inside the lumen. But if it says that it's going to be used within the cell, intracellular, then the ribosome will just stay where it is in the cytoplasm and produce the protein there. Cytoskeleton. And the cytoskeleton is sort of like one of those unsung heroes of the cell. It doesn't really get much press. Nobody really talks about it when we talk about organelles. But every single movement of the cell and every single movement that happens within the cell is either A, random, or B, decided by the cytoskeleton. Okay, so if the cell needs to move at all, which cells do need to move, even stationary cells that are part of a larger organism have lots of stuff inside that's moving around, and it couldn't move around if it didn't have the cytoskeleton. Okay, so that's its basic function, the cytoskeleton. That is a system of proteins, system of protein fibers. System of protein fibers that allows for all types of cellular movement. Okay, so there are two types of cellular movement. First one is called intracellular movement. Okay, intracellular movement means inside the cell. So if say a vesicle buds off of the endoplasmic reticulum and it needs to go to the Golgi apparatus, it needs to travel along a track of cytoskeleton, and we would call that intracellular movement because it's movement that's occurring inside of the cell. There's also just what's called basic cellular movement. Okay, and cellular movement refers to moving the whole cell. Okay, so if you have a motile cell, if you have one that has like cilia or flagella or something like that, the movement of the entire cell would be called cellular movement. Now, there's also what's called extracellular movement. Okay, extracellular movement is not actually controlled by the cytoskeleton. So extracellular movement, it happens in the extracellular space, and it's controlled by extracellular matrix, right, which is not part of the cytoskeleton. So, let's talk about the different types of cytoskeleton. The smallest type, we'll go there first, microfilaments. Okay, and you will hear microfilaments referred to interchangeably as actin filaments. And you hear them referred to interchangeably as actin filaments because they are made out of the protein actin. Made from the protein actin. Okay? And they're normally arranged in the exact same fashion, and that is two separate fibers that are bound together or wound up. Okay? So usually two fibers of actin that are bound together. Okay? They form like a helical structure because they're bound together. All right? They have two main functions in the cell. Their first function is for intracellular movement. Okay, so they're used for intracellular movement. And the way that that works is that they have these long fibers of actin, and then there is a smaller protein, and that smaller protein is called myosin. Okay, so myosin is used to move along the actin. Okay, and because of that, we would call myosin a motor protein. What's up?
So I kind of accidentally read the wrong pages last night. For the structure of microfilaments, it's like the globular actin that forms together and then twists and then bonds with like bonds or like joins with another one. That's doing the same thing.
Yep.
Okay, thanks.
Okay. So myosin is used to move along the actin. Myosin is what's called a motor protein. Motor just means to move, so this is a moving protein. Myosin is the moving protein because it moves. And the way that it moves is it has two different shapes. Okay, so myosin has two shapes. And one of those shapes is for when it has been phosphorylated, when it has ATP added to it. And the other shape is for when it hasn't been phosphorylated. Okay? So we call those the conformational shapes of myosin. But we're going to do a little drawing here. I'll show you sort of how it works. Okay, so this is going to be actin, right? The actin microfilament. It's a long strand, and there's two of them that are bound together, right? And then we've got say a vesicle has budded off from somewhere, so we've got a double membrane here. And then embedded in that membrane, you have these proteins, and one of the proteins could be myosin, okay? And so myosin can have two shapes. Let's say it's got this straight shape here, and then it's got the elongated bent shape like this, okay? And let's say that the elongated bent shape, that is what happens when you add ATP. So we're going to take an ATP, we're going to add it to this. Okay? So what happens then is this elongated shape is going to attach to the actin, and it's going to allow for this one to detach. We should probably label these red things. These red things are myosin. And this one is with ATP added. Okay? So we added ATP to it, and it changed its shape into this conformational change, this elongated shape. And then the other one can go ahead and detach then, and then it allows for this elongated one to then straighten out once the other one detaches, and that's going to move the vesicle in this direction, okay? And so then the next thing is you've got this straight leg now, and then this myosin will get ATP added to it, and it will elongate and attach again, and it's going to move down, sort of like walking or crawling. It's going to crawl down the actin filament until it gets to wherever its destination is. Okay? And we'll talk tomorrow, when we talk about plasma membrane, we'll talk about how it knows where its destination should be, and how it knows what track of actin microfilaments to get on. Yeah?
So, so do the different pieces of, so the different circles take turns being with ATP and without ATP?
Correct.
So how do they break the bonds of the ATP?
They break the bonds of the ATP. You mean once they've been phosphorylated, how do they go back to being non-phosphorylated?
Yes, how do they break those? How do they break those bonds?
It's the case that it is an unstable formation of the protein, and so that ATP that phosphorylates it and changes it only for a short time, and then it goes back to its normal structure because it's unstable. Right? So essentially the activation energy is less than the cell provides as thermal energy.
I haven't. Another question. How long would it take the vesicle to move across the cell?
Very, very quickly. And so, maybe if it was going from the endoplasmic reticulum to the Golgi apparatus, we're talking like a second or two, right? And so that means that when we're talking about these conformational changes of the proteins here, they're occurring really, really fast, right? You gotta think it's not that large of a distance to be traveling, but if you're talking on a microscopic scale,
what's the tallest you could be and still fit into a vesicle? Do you know?
No idea.
I was wondering if you could fit into a vesicle if you're 20 nanometers tall.
Fit into a vesicle? Sure. 20 nanometers isn't that much. All right. So, the other thing that microfilaments are really good for is that they provide an internal structure for the cell, and that structure is going to resist the crushing force. Okay, so they provide internal structure that resists crushing force. And this is, crushing force is referred to as inward force. Okay, so any pressure that's put inward on the cell is resisted by this internal structure here of microfilaments. Okay? Why would you have inward force? What's the main cause of inward force, do you think? Pressure. Yeah, the differences in pressure. So if you have, let's say you have a greater amount of solute versus the inside and the outside, that can cause inward force, or it could cause outward force based on if there was more solute on the inside, then you'd have more water trying to go in, and that would push the cell outward. Okay, let's talk about the next type, the intermediate filaments. Intermediate filaments are sort of like a catch-all. So there are lots of different types of intermediate filaments. It's not like just one type of protein, like in the case of a microfilament. Intermediate filaments can be many different proteins, and they call them intermediate because they're medium in size. They are larger than microfilaments, but they're smaller than microtubules. So medium-sized protein fibers of various types. And their main function, the main function of intermediate fibers, is to resist the shearing force, okay, or what's called the bursting force. So they resist bursting and shearing, which is called outward force. Okay, so for instance, your skin, if you take your finger and you rub your finger along another one, that applies a lot of force onto the cells of your skin, right? Your epithelial cells, right? But they don't burst, and the reason why they don't burst is because they have a whole bunch of intermediate filaments. Like if you went in to some non-epithelial cells in your body and you did the same movement there, they would burst apart in seconds, right? But that doesn't happen in the case of the skin cell because you have this intermediate filament in there called keratin, and keratin gives you really, really, really tough skin, right? It's the same thing that's in your fingernails, it's the same thing that's in your hair, right? Because your hair structure, it's also in your skin. It's the same structure that makes up the feathers of a bird and the scales of a reptile. Right? So keratin allows for cells to be extremely rigid and extremely resistant to this shearing force, this cross force. Yeah.
And do bacteria and prokaryotes also have cell skeletons?
They do, yeah. Yes. So that's a really good point there because you're saying prokaryotes, do they have cytoskeletons? They do have cytoskeletons, and they can have cytoskeletons because we said that prokaryotes lack membrane-bound organelles. But there's no membrane involved here, just protein structures. And so all the protein structures, or most of the protein structures, are present in both prokaryotes and in eukaryotes. Okay? Okay, so that's all we're going to say for intermediate filaments. We could go on and talk for days on that, talking about the different types, but we won't do that. So let's talk last about microtubules. The microtubules are the largest of all of the filaments. And these form hollow tubes of protein filaments. And because they form hollow tubes, we call the protein that they're made out of tubulin. So made from protein tubulin. Okay. One of the main things that they can do that separates them from the rest is that they can easily become shorter or longer. Okay, so they can easily shorten or elongate. And that's controlled by what we call microtubule control centers. Microtubule control centers allow for changes in shape. And the largest microtubule control center in the cell is the centrosome. Centrosomes are the largest control centers. Okay? And so not only can they easily shorten and elongate, but they work in the same way as sort of the microfilaments do, and that is that they have a motor protein. There's actually a couple motor proteins that are associated with them. We'll talk about that in a second, right? But that allows them to have a larger overall structure. So instead of just being one or two microtubules that are clumped together, you can have a whole bunch of microtubules that are clumped together into a much larger structure, okay? We'll say they can form large structures. Those large structures are things like flagella and cilia. Those are movement structures. Flagella are larger than cilia, and flagella tend to be fewer in numbers, and they move in two different ways. They can either move back and forth, sort of like the tail of a fish, right? And that can propel them, or the more common way that a flagella moves is it moves in a corkscrew, and that corkscrew will propel the organism forward. And then cilia are many little offshoots, and they sort of pulse like this, and when they're pulsing like this, that allows for them to create a current in the water, either to draw water towards them or to move through the water. Yeah?
Like how can we tell that they'll move in a spiral?
How can we tell?
Yeah.
They're large enough to see. The light microscope.
Is it how do we get to 3D view where you can kind of see that it's moving in a spiral, not just side to side?
Again, large enough to see. So the way that a light microscope works is that you're looking at one particular plane based on the focus that you have, and so you can see that certain parts of it are in focus and certain parts are out of focus because they're out of plane, and so the ones that are higher are out of focus and the ones that are lower are out of focus, and so you have a little bit of focus, out of focus, a little bit of focus, out of focus, a little bit of focus, out of focus, so on. You can tell that it's going like that. Anything else? Okay, so you can form flagella or cilia with them. Or you can also form what are called centrioles and basal bodies. Okay? And you're going to see this sort of concept repeated a whole bunch. So in this chapter, we're just going to see two different structures. So it's going to say, for the flagella, it's going to say 9 plus 2. Right? And then for the centrioles and basal bodies, it's going to say 9 plus 0. And what they're talking about is in the actual structure of these, there's going to be 9 bundles on the outside and then 2 bundles on the inside of these structures, or 9 bundles on the outside and 0 bundles on the inside. And so it looks like this. So you can see here, this is a 9 plus 2 pattern because you have 1 bundle, 2, 3, 4, 5, 6, 7, 8, 9 bundles on the outside, and then 2 on the inside. So they call this the 9 plus 2 pattern because there's 9 bundles on the outside and 2 on the inside. This is what you'd see on the flagella and on cilia, right? This is a basal body, also the same format that you have for the centrioles. Right, and you can see that they're 9 plus 0. So if we start up here, we got 1, 2, 3, 4, 5, 6, 7, 8, 9 bundles on the outside. These are in triplets, right? And then down here, you can see in the center, there's nothing. And so this is 9 plus 0 pattern, okay? And the way that this works is this is going to control the type of movement that these things have, okay? So let's talk about their movement here. So you've got a couple of different things going on here, right? So you can see, I wish the zoom wasn't so much in this. Reset to default. There we go. Good. So you can see here, this is the 9 plus 2 pattern. So you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, and then 2 on the inside. And then in between here, you can see these little red things. And this is the blown up version of it. Okay. These red things represent the motor proteins. Okay. And so these are a motor protein that's called dynein, right? But it could be a number of other types of motor proteins that are associated with microtubules as well. Okay, so I'm gonna hit replay here, and we're gonna listen to this for just 2 seconds or a minute, for however long it takes.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella, or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP. Click on the microtubules to see how they work in a flagellum.
Okay, so here you've got these microtubules here, and each one is connected to the next one by these motor proteins, okay? And so if you were to have those motor proteins flex, what you'll see is a shortening of one side of the microtubule. And if you have a pattern of flexing of 1, 2, 3, 4, 5, 6, 7, 8, 9, then what you'll get is a corkscrew style movement, right? If you have the flexing of one whole side, you have a back and forth movement, right? So the way that these motor proteins function is going to allow them to change the movement. So if you look here at these motor proteins, right, what happens is you phosphorylate them. When you phosphorylate them, they change their shape, and that allows them to bend and attach to a different spot. And then when they go back to their non-phosphorylated shape, that allows them to shift where they're attached to the next microtubule. Does that make sense? Everybody see that? What's up, George?
It says that it's 9 surrounding pairs plus a single central pair. So how come it's 9 plus 2 and not 9 plus 1?
Because they name the central pairs, or the central tubules, not based on pairing, but based on the total number of them. And then the outsides can be triplets, or they can be pairs, or they can be quadruplets, based on the structure. Yeah?
Okay, so flagella and cilia can both be used for movement.
Mm-hmm.
And then cilia can also be used on stationary cells, like operate in water.
Sure, yeah. So if you think about a cell, you really have 2 different methods for getting to food, right? Method number 1 is to actually move yourself to the food. Method number 2 is to move the food to you, right? And so if you have cilia, you can either use that to move you to the food by moving the whole cell, or you can create like a circular pattern, right, with the cilia. And if you create a circular pattern, what that does is it creates like almost like a drain or a whirlpool effect around the cell, and that draws things towards it, draws the food towards it. So the cell itself doesn't have to move, but it just pulls things that are around it. Flagella can do that as well, but it takes many more of them, and generally you only see like 1, 2, or 3 flagella on a cell, whereas you see hundreds of cilia. Yeah. Sure. Does anybody have any other questions on the cytoskeleton? All right, you got like 3 minutes left.